you have tons of publications, and I was want to talk about one specifically the genital microbiome and its potential for detecting sexual assault because I actually do a lot of sexual assault cases at my job here. So could you give us a little overview in that? And I know you and Nitty are good in the DNA and genetics department, so if you could dumb it down for me, please.
Yes, yeah, that was one of the very interesting pilot studies that we actually did. So we all know that we have like a backlog in sexual assault cases, and the
Right.
amount of cases where they're either not tested or the ones that are tested and you don't get a full profile or not even a partial profile that would be eligible for CORIS and to continue with the conventional method. And those cases oftentimes are considered cold with no closure for the victim whatsoever. So we started thinking of alternative methods of potentially at least proof objectively if a sexual contact occurs. And that's where the concept of the natural bacteria that are present in the genital area for females and males, where the interest. So we're like, are they different? If they are different, which bacteria that are present there can distinguish a female from a male? Are they specific just to the genital area? Because based on the Locard principle of exchange, that anything that come in contact should transfer something. And because we have so many bacteria compared to how many human cells, the idea was like, the hypothesis was like, okay, if I don't find a foreign male DNA, am I still gonna find at least some male specific bacteria that can prove if a sexual contact occurs? And that's where this project all came along.
Okay.
So is that the step you take after you look for, after the screening is done for a sexual assault and you can't find sperm?
Correct. So the thought is, obviously now it's not in any actual casework. It's still in the research phase because we still need to understand a lot about the transferability, stability, the signature, and all of that. But yes, the idea would be eventually that when you don't have male DNA, because that's the gold standard. Obviously, you want something to identify a specific perpetrator, try to find a match in the database, and go all the conventional method, do all the statistics. But in cases we're saying, okay, we don't have enough male DNA, can I still exploit that same sample that I'm collecting from the sexual assault kit and look for those signature bacteria that are specific?
But then if you, sorry.
Yeah.
So you don't have enough male DNA, so you can't do the test came out inconclusive for male, or maybe no male DNA, human sperm present. Then you move to gut. I'm just thinking of the pipeline. Then you move to knowing if using the gut, using the microbes to see if genital microbes to see if male is present. But then if you still have to get a profile for DNA, because you can't just rely on that because they are probably not confirmatory because it would be the same across all humans. So
that's actually a very good question. And then that's where there is a need, obviously, for more research. So what so far we know is that there are bacteria that are, the ones that are for female are way more studied and understood. But even if you think of us one bacterial species, when we did now a continuation of the study that you're just talking about that was on couples pre-coital and post-coital, so before sexual contact and after sexual contact. And you would think, let's say bacteria A is present in 2 couples. If you look at the SNPs or the polymorphisms within these bacteria, you're gonna even see further differences. So even if it's the same bacterium, it's just the family or the genus, when you look at the species and the strain level, even the strain level, that can show you some SNP differences that can differentiate the same bacterium from one individual to another. Obviously, we have to do more population study to see how strong of an evidence it could be, whether it can ultimately be confirmatory or maybe presumptive, but can solve some cases where, the victim is a child who cannot speak for themselves, or an elderly who cannot speak for themselves, can you at least prove that, yes, indeed, a sexual contact occurred, even if you cannot probably pinpoint it to a specific person, but narrow it down to a potential pool of suspects?
And then you can also use single cell technology if it is
There we
present.
go. We complement our
So you never
research.
know. I'm sorry, I tried to push in whatever
Yes.
possible what I
We
do.
all, as researchers, that's what we want to do, show the pros and cons, or the advantages and disadvantages of every technology and every methodology, and whether there is a need to combine, if possible, different methodologies for more power of this discrimination.